Hey, good morning, everyone. It's been nice to see such a good turnout. Two really excellent talks so far, and I know the third one is going to be excellent. So I'm pleased to introduce Dr. Ray Wheeler. I've known Ray for a long time. He's a plant physiologist, and he's the lead for the advanced life support activities at Kennedy Space Center. He began his career at NASA in 1988, and he's studied hydroponic techniques for space farming experiments, expert in bioregenerative life support, plants and bioregenerative life support. Ray is also very active in our society over many years, and he is a recipient of the highest award, the Founders Award. So Dr. Wheeler, please, thank you.
Thanks, John, and thanks to the Fellows Committee for this first year to participate in this. So I'm going to talk about bioregenerative life support. So hopefully you'll understand what I mean with that as I go through some of these slides. So if you think about keeping humans alive in space, you can look at these kinds of diagrams, very simplified, but we need food and we need oxygen, and then we generate waste products like carbon dioxide, and we need clean water, and we make wastewater. We're good at that. If you use photosynthetic organisms like plants, you can drive that reaction in the other direction, but you need a lot of light energy. As shown in that bottom diagram, the plants or other photosynthetic organisms can take the carbon dioxide, they split the water photolytically, that's where they get their electrons, and that releases oxygen. So that's a waste product, but that's something that we need. So, and then if you choose crops, for example, that biomass that you generate is going to be food. So you can see there's a nice multiple kind of life support function by using organisms that are photosynthetic. So this is a form of bioregenerative life support. You can extend this to waste processing and many other aspects of keeping humans alive in space. So what would we do on the moon? And you see diagrams like this. I like this one. This was developed by a group at the University of Wisconsin in the early 1990s, and you see maybe a moon colony there, and they've heaped regolith over their living quarters for radiation shielding. And then one of those sections is, you clearly see there are plants growing there, you know. And I'm convinced they're potatoes. I used to study potatoes, so I know I can see that very clearly. But, so things like that. That won't be the first step, though. We're not going to, right out the gate, going to build a big colony like that and have set up a big space farm. So how do you get going with something like this? And so it really needs to be a sequential development, in my opinion. And others have talked about this for many years. Maybe you begin to just provide some fresh, perishable foods to the crew members, things that don't store very well, but that can then help them in their diet. And then you follow this evolutionary path and you expand things as you have the opportunities to do so when you get to a destination and stay there. We do know that without providing a lot of oxygen and removing CO2, just with a few, maybe a half of a square meter of growing area, we can grow a lot of supplemental fresh foods, things like leafy greens, could be strawberries, radishes. And so these could be very important for adding some key nutrients to the diet on a sustained basis. For example, we know there are some vitamins that don't last long in the packaged foods. They just have a shelf life. Things like vitamin C and vitamin B1. Well, if you could grow plants, for example, that could provide these on a sustaining basis, you could meet these kinds of nutritional gaps. Plus, you can see there's color, there's texture, there's aromas. So this adds to the whole experience of eating food. And so that's important, but it's a difficult one to quantify. When you do trade studies with engineers, they want to know your mass and your power and your volume and so forth. When you say, hey, this is a good thing to have, they'll say, give us a number. And so that's where we need a little bit more information to quantify that. Turns out we're doing this already. We've taken this first step. If you look at the International Space Station, orbiting in low Earth orbit, there are several plant chambers going there right now. NASA operates some of these. The Russians have operated a chamber there. It's no longer there. It was called LADA. But so there you see a picture of Peggy Whitson tending some Chinese cabbage plants in the veggie plant chamber. And on the right, you see some pak choi plants that were grown there. They were one of the better ones that we did. I see Joya nodding, yeah. And then you see the crew actually eating and enjoying some of this. They were consuming wasabi mustard greens there, and you can see the one astronaut maybe took a little bit too much, had a mouthful of nice, hot, spicy mustard. So that's in a space station. What happens when you get to a surface setting, like the moon? So you're going to have a small habitat that's going to be very crammed and full of things. Is this an opportunity, maybe, to begin growing small amounts of plants to test these kinds of concepts? We think so. This is a picture of what was called the Habitat Demonstration Unit. It tested for 3 years. 2 of them were at this desert test site in Arizona near Flagstaff, and then 1 at Johnson Space Center. That chamber is now the Hera chamber at Johnson Space Center. Some of you might have heard about that. But anyway, we thought, well, hey, let's get in this as plant biologists. Let's get in on the action. And the first year we had a whole shelf to ourselves, and then later on all kinds of groups came in and they said, you guys, there's no more room. So we had to get a little creative, and so we got them to design a shelf around the ladder going between the upper and lower portions of the habitat. The living quarters were up above, the sleeping quarters, and then the lab was down below. So we had this, we called it the atrium, and you can see that we grew plants there. And we allowed the crew to tend those, and they even ate some of these fresh foods that they grew there. Well, the concept of using agriculture, bioregenerative systems is not new. It's been talked about ever since space programs existed. And in fact, there was a book written on this by Doug Ming, a geochemist, and Don Henninger, a soil scientist. They edited this book, and it talked about lunar-based agriculture. And it had lots of interesting chapters in it, some talking about maybe using regolith to grow plants, others talking about hydroponic systems, bioregenerative life support, and so forth. So nothing new here, and people have been thinking about this for a long time. I think a logical place would be probably starting with hydroponic systems, not maybe of the scale you see there in that left-hand picture. That's a deep flow system at Cornell University. But hydroponics should work under 1/6G, I think. Water should flow downhill. We can use gravity to a mechanical advantage. Usually in recirculating hydroponic systems, you add water at one end and then it runs downhill. You collect it in a catchment reservoir or a sump, and then you keep pumping it around the loop. Or you could do it in smaller trays or troughs that are shown there on the right side. So I think small-scale hydroponics might be a good starting point. Maybe we do use regolith over time. As we process this, this is the soil rubble or the material that exists on the surface. We just heard from Clive. And there are a lot of elements there that plants do require. On the left side, I've showed the list of essential elements for plants to grow, kind of in descending order there. And on the right is one example of some analysis of samples that were returned, in this case, Apollo 15 site. There are others of these. And these are all expressed as oxides. I don't know why geochemists do that. Clive, why do they do that? Anyway. It's easier. Okay, there you go. So, but if you look, there are some essential elements right there for plants. But there's no nitrogen, there's not enough potassium, so we'll need to fertilize, bottom line. Even farmers that have great field soil in Illinois and in Iowa, they still fertilize their corn with nitrogen. So you're going to need to do that with regolith. Another thing, if you do think about regolith, for growing plants. You need to think about the particle size. This is from a paper by McKay and Ming, and it shows a distribution of particle size. Don't worry about reading it. There's lots of fine particles in the surface regolith, really fine stuff. And so under 1,6G, when you add water, and here I've done a kind of a diagram of pots under different gravities, and on the left you see, say, a fine material, maybe about a sand-sized particle, and under 1G you add water, you let it drain, and then you get a wetting profile. It maybe looks something like that. It tends to be a little more wet at the bottom. You get sort of a perched water table. Under a Mars gravity, it doesn't drain as far for that given distance. And under lunar gravity, you might get very little drainage at all. And so this is a physical phenomenon now that you need to consider if you're thinking about regolith as a growing medium for plants. You can get by that, though, if you increase the particle size. And here I'm drawing on some papers by Scott Jones. He's written a lot on this. Scott Jones is at Utah State. He's a soil scientist. So if you have larger particles and bigger pores, you can now get that kind of water distribution profile you see under 1G, even under reduced gravities. You can't have any fines, though, stuck in between those big gaps because it won't drain. So this kind of complicates things if you think about using regolith under reduced gravity. But the good side is it provides a three-dimensional root zone. It provides anchorage for crops that maybe grow taller, maybe dwarf fruit trees, things like this. So, something certainly to consider. So what about crop physiological responses in reduced gravity? The 1,6G, for example, on the moon. I think, and from some great talks we heard yesterday from John Kiss, Chris Wolverton, and others, I think real sensitive responses like young grass coleoptiles and first true leaves or hypocotyls of dicots or primary roots, these are really sensitive organs. And so I think 1,6G will be enough to orient them and tell them which way to go, which way is up, which way is down. We don't know for sure, but I think it's a pretty good bet. We know from studies that have been done in space and simulating fractional G. What about other organs, though? Plants respond to gravity in all kinds of ways. So I was walking through my yard and I took a picture of this banana inflorescence, and you see in the upper left there, bananas have this structure called a bell, and it hangs down, and it's a heavy structure, and it acts kind of like a plumb bob. And so it pulls the fruit cluster down, and then as the fruits grow, they grow in a negatively gravitropic fashion, upward. Will 1,6G be enough for a banana? I don't know. Maybe they'll be confused. Who knows? Peanuts are another interesting species. Some of you might remember Lou Feldman at Berkeley studied peanut gynophores for many years for NASA. So peanuts are a legume. They flower above ground. When that flower gets pollinated and makes ovules, there's a structure called a gynophore or a peg that then goes straight down the plumb line. So it's a shoot organ that's positively gravitropic, which is really weird. It's more like a primary root instead of a shoot. But once it gets down and then it does its thing. Will peanuts know what to do under 1/6 gravity? Don't know. That's a pretty strong response. I bet you they would. What about plagiotropic organs? That refers to organs that go at some angle to the plumb line. So in the lower right-hand picture are some potatoes that I grew when I was at the University of Wisconsin, and I dug them up. And so you can see these underground, what potato growers call stolons. They're rhizomes, but they're underground stems, and they naturally go at an angle to gravity. It's not that they're confused. They know exactly what they're doing. And we've done time-lapse photography of this, and you can watch stolons of potatoes circumnutating almost around a direct horizontal line. Will that be enough stimulus in 1,6G to keep potatoes happy? Will they be confused? I do know that when potatoes aren't completely induced to tuberize, some of those underground stems will pop up. They'll become positively gravitropic. So there's an interesting, maybe potential connection there between the induction factor of tuber formation and gravity. And then John mentioned the other day, plants are assimilating all kinds of environmental stimuli. And so in that upper right-hand picture, I showed some Phalaenopsis orchids, and they all have an inflorescence spike on them, and they're all headed off to the left, you can see there. And that's because there's shade cloth right above them in that greenhouse, and there's more light off to the left. And so they're all using phototropic orientation, and this is under 1G. But just to show an example of why plants have to kind of integrate all their stimuli, and if the gravity is reduced, how will this affect things? And there's other things. Will apical dominance, that means when you have a stem, a shoot that's dominant, will that stay normal under reduced gravity? Don't know. Will you get compression wood and tension wood? I was asking Norm Lewis the other day. He said, well, yes, it can happen even in microgravity if you bend the stem. Do we want compression wood and tension wood? I don't know. Grasses are interesting. Some of you might remember Peter Kaufman at the University of Michigan. In the 1980s had NASA research grants, and he studied grasses, and they have nodes or pulvini structures. And so when a grass stem falls over, these quiescent nodes, if they're still viable, will begin to grow, and they'll turn the knocked over stem upright. It'll kind of be like an elbow bending upward. So if the grain grass, for example, gets lodged, it can self-correct. Will that be enough in 1.6G? We don't know. Okay, well, whatever we're doing with plants, we're going to need a lot of light, especially if you want to have a productive system. And if you're using electric lighting, LEDs are now the hands-down winner. 20 years ago, this wasn't the case. We were all using high-pressure sodium lamps. LEDs have just improved remarkably. It's astonishing to me. They're now 70 to 80% efficient. If you put 100 watts of power into a state-of-the-art LED, you're going to get 80 watts of light out. I mean, that almost violates a law of thermodynamics to me. But they're going to be the way to go. And by the way, I don't know if you can read one of the footnotes there, and Alan, this is something to point out. The use of LEDs to grow plants was patented through NASA funding to a commercially funded group at the University of Wisconsin. This was a Code C center called WIXAR. And Bob Morrow, if you're in the audience somewhere, Bob is one of the co-signers of that patent for using LEDs to grow plants. And everybody's using LEDs to grow plants. So Bob is fabulously wealthy now. And if you see him in a bar tonight, ask him to buy you a drink. Well, maybe you aren't that wealthy, Bob. I don't know. Maybe you need to buy Bob a drink. Yeah, buy Bob a drink for coming up with this. But we can use sunlight. Depends where you go. Maybe you collect it with things like these concentrators that we tested at Kennedy Space Center. We had this collection system on the roof. You see these parabolic mirrors. They would then reflect light to I don't have a pointer. But take my word for it, there's a secondary mirror at the top of that. So it's like a reflecting telescope. And this was a dichroic mirror as well. So it let the long wave radiation through, reflect the light back on a fiber optic bundle, and that would then be conveyed down into the building, into a plant growth chamber. And then these would track the sun through the day. So maybe you could use this principle, or you could even use Fresnel lenses, a magnifying glass kind of principle. So that's a refracting telescope in that case principle. So we heard a lot about Shackleton Crater in the South Pole of the Moon. I pulled out some old charts from the Constellation program. We're going to go there then, too, Clive. That's one thing I've learned with NASA. Save your old road maps and charts. They'll come back. You know, just put them in the file for a while. So here's the edge and the rim of Shackleton Crater, and you can see there are zones there where you have greater than 75% light, maybe almost permanent light. And as Clive mentioned, you're only kilometers away, but I didn't realize how steep the slope is from permanently shadowed areas. But there's resources nearby, and there's light there. So if you're parked up on top, you could use solar light. And here's a diagram that ESA came up with showing a greenhouse kind of structure, and I like this round hemispherical structure. It could hold pressure, and then if you're at a high latitude, the sun's going around low on the horizon, and that'd be a good way to capture light, perhaps. Transparent structures have lots of challenges. You got to hold pressure, the materials have to hold up to the intense UV radiation. You have thermal issues, but maybe we'll come up with some materials. People have even talked about deployable structures that could be used for greenhouses, and this is a concept developed by the University of Arizona. It's a collapsible structure, a greenhouse-like structure. It kind of collapses like a slinky, and so they can expand it out, and then they can fill this with plant growing systems. And in this next picture, if you look inside, this is another view. They even came up with a concept where the hydroponic troughs were made of flexible plastic pouches, so they could be collapsed as well. So now you don't have any rigid plumbing that you have to deal with. I thought this is a very clever idea. So the whole system is collapsible. I'm running out of time, so I better keep moving here. I want to make a point about ground analogs here and the value of them. We had a ground test facility at Kennedy Space Center many years. This was called the Biomass Production Chamber, and we had stacks of shelves of hydroponic systems and lights and hydroponic shelves and lights and so forth. And at the time, we didn't know this. The term didn't even exist, vertical agriculture. I think this was probably one of the first working examples of that. But we don't have that anymore. ESA has a test bed for bioregenerative life support. This is at their pilot plant in Barcelona. The Japanese in Amore Prefecture had a very elaborate test facility. It's now being used for other purposes, for isotope tracing, but they used it for life support, growing plants and bioregenerative. The Chinese are very active in this area. This is a group at Beihang University in Beijing. And so they've enclosed humans. They've done human studies up to a year where they've kept them alive with plants and bioregenerative systems. The German Space Agency has a module in the Antarctic as well. Rob Furl and Annalisa Paul, probably in the audience, or certainly here at the meeting, have been participated in this, as well as one of our colleagues at Kennedy Space Center on the left there, Jess Bunchek. She's there right now, running this plant chamber. NASA's current analogs, nothing. People often ask me these kinds of numbers. This is all dependent on light, but if you have something like 20 to 25 square meters of crops, you can produce enough oxygen for one person. So you're continually planting, harvesting, and that removes their CO2. If you double that number, that depends on the harvest index of the crops, you can probably provide enough dietary calories. And plants evaporate lots of water. You can generate lots of clean water with plants. There's 2 orders of magnitude more water vapor moving through a plant chamber than CO2 or oxygen. But this depends on body mass. Honestly, if you choose people with lower body mass, you win across all of these life support functions. You need less oxygen, you need less food, they generate less CO2. So bioregenerative systems, we need them on the Earth. That's what's keeping us going right now, and we're doing our best to damage these systems. And I'm convinced we'll need them in the future as we continue to explore space. And let me just close with this one slide in tribute to my academic advisor, Frank Salisbury, who talked about this very theme 30 years ago when he addressed the American Society of Horticulture. Frank was a longstanding member of this society. He did both fundamental gravitational biology as well as agriculture, space agriculture work. So thank you all very much.
Well, unfortunately, thanks to earlier delays, we're not going to have question time because the education group will come in. But that picture up there, everybody should read that article. It is really quite impressive by Frank Salisbury. That being said, I should point out that all 3 of this morning's speakers did a lot of homework for this session, and I think we need to thank them all, not only for their skills as orators, but the effort that they put in for us. Thank you all.